Well, hello everyone, and welcome to Introduction to Psychology. We have our next chapter here, which is going to cover the smallest part and the most foundational part of who you are. I'm really excited about this time together and how we're really going to appreciate how, even at the very small level of who you are, you're amazing. So this chapter is going to cover the biological basis of behavior. So we're going to be talking about neurons and areas in the brain and processes in the body. I don't want you to lose sight of the fact that this stuff is amazing. I don't want you to just memorize a whole bunch of brain parts. I don't want you to just be bogged down by details. I want you to be excited about details and amused and amazed by the structure of what makes you you. So I'm looking forward to this chapter. I'm looking forward to teaching it to you. And wherever this week finds you, I hope that this material challenges you, excites you, and gives you a bigger picture of how psychology is trying to answer the question, why do people do what they do? So in this chapter, we're going to go to the smallest, most micro place that could answer that question, and that has to do with your biology. This chapter is going to try to explain the biological mechanisms that underlie that human behavior and mental process. These physiological and anatomical foundations are the basis for many areas in psychology. So in this chapter, you're going to learn about genetics and physiology and psychology. You're going to learn about neurons and brain regions. And hopefully it's going to give you more foundations for understanding why we do what we do. You're going to see that this system that you have, your brain, spinal cord, nerves, endocrine system, and multiple systems in the body, we're talking about billions of cells that communicate and integrate and consolidate and interact in such a way as to make you be able to be yourself. Whether that is studying for a test, or driving a car, or singing a song, or listening to this lecture. So in all, what is this chapter going to cover? Well, we're going to talk about the nervous system. So we're going to talk about the central nervous system and the peripheral nervous system. We're going to talk more specifically about neurons and how special they are as a cell, how amazing they are, and how foundational they are to your behavior and mental processes. We're also going to talk about the brain, which is totally amazing and complex. And there are so many journeys and adventures and findings about the human brain that we haven't even gotten close to yet. So we're going to discuss a little bit about what we know presently. We're going to talk about how we know certain brain parts are involved in certain functions. So that has to do with damage and repair and all of the ways that we try to scan and look at the brain. And near the tail end of the chapter, we're going to talk briefly about genetics and briefly about the endocrine system. So let's get started. So there are many different approaches to psychology. As you know from chapter 1, every approach has a different lens through which it looks at human behavior. In the biological approach, we're going to look at the smallest pieces, the cellular pieces, of who you are and why you do what you do. Learning how the body's cells and organs function can help us to understand the biological basis of our human psychology. Broadly speaking, in biological psychology, many researchers will study the function of the nervous system. That's brain, spinal cord, and nerves. This is a field called neuroscience. And it studies essentially the body's electrochemical communication circuitry. And each word there is important because it's indicating two important pieces of how you function. There is an electrical part of the nervous system, meaning that there are impulses and electrical charge, and also chemical processes that have to do with hormones, neurotransmitters, and the chemical nature of who you are. And who you are in this nervous system circuitry is an electrical chemical system. Your book makes mention of the fact that there are billions of neurons in your brain. Billions! I mean, imagine this. And they're so densely packed in the area in which they're placed that they're one of the most amazingly interconnected systems on Earth that we know about. So even if you're having like a really bad day and feeling really bad about yourself, you just need to remember that your brain is one of the most fascinating things in the solar system. It's amazing. So let's talk a little bit more about the complexity of your nervous system and how awesome it is. Your nervous system absolutely is complex and complicated. This means that it has multiple inputs, multiple outputs, and multiple connections. This means as a system, it is functioning on multiple levels. It is not just an A plus B. It's an entire song and symphony of complexity. It's also integrated, and that means that there are interconnections between the brain and body, and the body and the brain, and the brain and the brain, and the body and the body. And all these interconnections are meant to consolidate, connect, and essentially create a fluid psychological response, whether that's a behavior or a mental process. Your nervous system is also highly adaptable. We use the word plasticity to mean that it can learn and grow and repair. That plasticity is something that is tremendously at the forefront of neurological and neurobiological research. And as I mentioned, this process is not unidimensional. It is electro and chemical. So when you take that all together and you imagine that every neuron cell is connected with nearly 10,000 other cells, that is a complex system. When you think also about the fact that your brain has the ability to heal and restore and even recover from damage, and that it rewires itself in accordance with the experiences and habits and thoughts that you have. Wow, that's amazing. One of my favorite neuroscientists, his name is Daniel Siegel. He's from UCLA. I use his book on parenting in my child development classes, so I want to fangirl about him for a little bit. But he talks about how amazing this integration process is. That who you are, and on the biological level, on the brain level, is desirous of making sense of experiences. In an ever-moving world with challenges all around you, your brain wants to make sense of logic and feeling. It wants to be able to deal with basic drive as well as higher order cognitive processes. The application here is that it's so interesting, especially early on when the brain is especially plastic and moldable and teachable, is that the sensations that you have, the images in your mind that you have, the feelings that you have, the thoughts that you have, and the behaviors in which you engage, they strengthen connections in the brain. And your brain goes in the direction of what it's done before, in such a way as if you think in a certain way, your brain starts to strengthen thinking in that way. Or if you engage in a habit, that behavior becomes something that becomes stronger and more motivated in the brain the more you do it. What's the application here? All of us have things and ways of thinking that maybe aren't as healthy or adaptable as they could be. But fear not. Whether it's a habit or a negative thought that you consistently have about yourself, your brain wants to reorganize, and it has the capability of doing so. You just got to give it the input to do so. It's been my experience that students like you guys are so hard on yourself, and you have a lot of negative self-talk. Well, be careful, right? Those thoughts, every time you have that thought, a neuron that fires together wires together. They become better friends neurologically. They become more connected. Their connectivity is stronger. So if you want to do something new, you got to do and think something new. So let's talk a little bit more about the pieces of the nervous system. The nervous system is composed of two basic cell types, glial cells and neurons. So glial cells are essentially like supportive. They have to do with cell upkeep, nutrition to the cell, repairing cells that are damaged, and supporting pre-existing cells where they're placed. This supportive role is both a physical and a metabolic support. So they help neurons line up closely with one another so that they can communicate, so that there can be neuronal communication. Glial cells provide insulation to neurons. They transport nutrients and waste products into and out of neurons, and they help mediate and be part of the immune system response. And there are so many glial cells in your system, but it looks like there are nearly 10 glia or glial cells for every single neuron. So your nervous system has glial cells and neurons. And in regard to neurons, you're going to have afferent, efferent, and interneurons. So let's describe each of those. Afferent nerves are essentially bundles of neurons. So a nerve is a bundle of neurons. The neuron is the individual cell. The nerve is multiple neurons all packed together. So when we say afferent nerve or afferent neuron, what we're meaning is essentially a sensory nerve. In the afferent circuit, what's happening is external or even internal body processes. That information is going from the outside or inside of who you are and transporting itself to the brain and spinal cord. So it is accessing the brain and spinal cord. Efferent neurons or nerves, these are motor nerves, and they essentially exit the brain and spinal cord so that that information can go from the brain and spinal cord to the outside areas of the body, fingertips, digestive tract, toes. So when your hand touches a hot stove and the signal from your fingertips goes up your arm to your brain, and your brain goes, oh, I'm touching a hot stove. That's an afferent pathway. It is accessing or arriving at the brain and spinal cord. But when I say ouch and I move my hand off of the hot stove, my brain and spinal cord have had to tell my arm and my nerves and my muscles and the lips and my vocal cords to respond. So an efferent pathway means that my brain and spinal cord are saying to my fingertips, move those, and saying to my lips, move those too. Ouch. Afferent accesses the brain and spinal cord, and efferent exits from the brain and spinal cord. You also have a system of interneurons, or neurons that interface between those two pathways. And these pathways carry important information about the external environment, the internal environment of who you are, and how you should behave in a world that's constantly moving around you. All right, let's talk a little bit more about the nervous system. So we talked about nerves, afferent, efferent, and interneurons. We now want to talk about the main divisions of your nervous system. So the nervous system can be divided into two major subdivisions, the central nervous system and the peripheral nervous system. The central nervous system is comprised of the brain and spinal cord. And almost all of the neurons in your body, around 99% of the neurons that you have, they're in this central nervous system. The peripheral nervous system connects the central nervous system to the rest of the body. So this peripheral nervous system, it's made up of sensory and motor nerves, afferent and efferent, right? And it helps the central nervous system carry out actions, responses, adaptations to the environment. So let's discuss the peripheral nervous system. The peripheral nervous system is made up of thick bundles of axons, and we call those bundles of axons nerves. And they carry messages back and forth between the central nervous system and your muscles, organs, and all of the senses that are in the periphery. That's why it's called peripheral. Everything essentially outside of the brain and spinal cord. The peripheral nervous system, as you can see on this slide, has two major subdivisions: the somatic nervous system and the autonomic nervous system. So the somatic nervous system is associated with activities that are traditionally thought of as conscious or voluntary. It's involved in the relay of sensory and motor information to and from the central nervous system. And you know where we're going here with this. So it consists of motor neurons and sensory neurons. Those motor neurons you already know are efferent, meaning exiting the brain and spinal cord. And those sensory neurons are carrying information they're accessing, they're arriving at the central nervous system. So that somatic system has both afferent and efferent fibers. So each nerve is essentially a bundle of neurons containing thousands of axons, both efferent and afferent. So that is like a superhighway where information can go in the two directions, because you have the two different types of neurons. The peripheral nervous system is further divided, so you have somatic and autonomic. And the autonomic nervous system controls your internal organs and your glands. And it's generally considered to sort of be outside the realm of voluntary control. And essentially what we're seeing is messages to and from the internal organs: breathing, heart rate, digestion. The autonomic nervous system can be further subdivided into sympathetic and parasympathetic divisions. The sympathetic nervous system has to do with preparing the body for stress-related activities or survival activities, whereas the parasympathetic nervous system is associated with returning the body to a routine, day-to-day sort of state. These two systems are complementary. They operate in tandem to maintain your body's homeostasis. Homeostasis, as you know, is that balance in which the biological conditions of your body are maintained at optimal, healthy survival levels. So the sympathetic nervous system is going to act to prepare your body for fight, flight, or freeze. It needs to survive. You'll see that system is going to dilate your pupils, increase your heart rate. It's going to inhibit salivation. It's going to inhibit digestion. It's going to dilate the bronchi in your lungs and essentially give you more air to breathe. If you have to fight, flight, or freeze from a threat, the sympathetic nervous system is amping your body up for protection. In an opposite manner, the parasympathetic nervous system is going to do those, as your book says, rest and digest functions. It's going to help the body return to a daily functioning state, just the normal operating procedures, normal stuff. It's going to constrict your pupils. It's going to stimulate salivation. It'll slow your heart rate down. It'll get your lungs into a more easy breathing pattern. It's going to stimulate digestion and those processes that need to go on, and basically return your body back to a normal homeostatic state. As you can see and imagine, the sympathetic nervous system is activated when we're faced with super stressful or high arousal situations. All of those sympathetic nervous system responses, they are a constellation of physiological changes that allow the body to access energy and energy reserves in a heightened state of protection so that there can be fight, flight, or freeze. You can see that the system is protecting itself. And there's a great connection point with the end of your chapter. At the end of the chapter, there's a discussion of stress and how your system, when it's stressed, it's not good for this system, this sympathetic nervous system, to be aroused and activated all the time. What happens is this sympathetic nervous system response, if it's chronic, it's very costly. It takes a lot of energy for the system to run high on stress and high on arousal and high on the sympathetic nervous system response. If you're stressed for prolonged periods of time, the other systems that connect with that, your immune function, your endocrine function, your cognitive function, those things are going to be damaged. In fact, we suffer many negative health consequences when faced constantly with psychological threats that we can neither flee or fight. So chronic stress is associated with an increase in susceptibility to heart disease, as well as impaired function of the immune system. And those are just two of the negative consequences of persistent and repeated exposure to stressful situations. Thankfully, the parasympathetic nervous system wants to restore function. It wants to return the body to a relaxed, functioning, healthy state, because those restorative processes are its responsibility. So what's the life application here? Well, the life application has to do with the fact that if you run really high on stress for a really long time, you could make yourself sick. We're all in a new situation here. It's a new season of life. So never begrudge the fact that self-care and allowing your parasympathetic nervous system to do its job is a good thing. Get some good sleep. Eat some vegetables. Do some stuff that returns your body to its restorative resting state. That's good for you. Because this college thing is not just a sprint, it's a marathon. So you have to take care of yourself as you do this marathon race. All right, moving forward, we're going to return to the two basic foundational cells of the nervous system. We have glial cells and neurons. We want to focus a little bit more on neurons now. So for review, glial cells have to do with supportive, restorative, protective functions in the nervous system. There's 10 to 1. There's just a lot of glial cells to each neuron for structure, support, nutrition, repair, and essentially like immune response and allowing the body to respond quickly. Neurons are amazing. Neurons are the other cell in the nervous system, and you have about 100 billion just in your brain. 100 billion neurons in your brain. And each of those neurons connects with around 10,000 other cells. Most neurons are created very early in prenatal development, but they do, in their size, shape, connection, and function, change over the lifespan. We call that plasticity. Neurons are the electrical chemical messengers of information processing in your nervous system. And those neurons are either sensory, afferent, motor, efferent, or interneurons, kind of communicating between systems. But there is a special type of neuron called a mirror neuron that is both motor and sensory. It's amazing. It has a dual function. We know about mirror neurons because in the late 90s in Parma, Italy, there was a researcher, neuroscientist, doing research on macaques and monkeys. This researcher by the name of Rizzolatti noticed that when the monkeys were observing other people in the lab eating, the areas in their brain that would have to do with the motor function of bringing food to the face and that motor action of putting their hand to their face, that those areas in the brain were lighting up even though their arm was not coming to their face. So what does this mean? You have your little monkey and he has electrodes in his brain. He's not moving. He's just watching the world around him. And the brain is watching a researcher eat a sandwich or drink a drink. And the area of the brain that has to do with intentional action of putting the hand to the mouth, those areas are lighting up even though the monkey is not actually doing that. This discovery by Rizzolatti, because of this research, allows us to know that there's a special type of neuron called a mirror neuron. And a mirror neuron is a neuron that fires both when an animal acts and when the animal observes the same action being performed by another creature, another person, another animal. This is why we call it a mirror neuron, because the neuron mirrors the behavior of the other person, as though the observer were acting and doing the behavior themselves. We're just starting to understand some of the implications of these specialized cells. They have great implication for social behavior, social thinking, what we call social cognition, social perception and engaging in social settings, the connection that happens when people mimic one another or imitate one another, and even bigger things like empathy and social skill. The science is still developing our knowledge of what these really awesome cells are for and their function and their action. All right, so I want to take our discussion back to the basic structure of a neuron. So let's now talk a little bit about neuron structure. Neurons are the central building blocks of the nervous system. They are the rock stars of the nervous system. And you have nearly 100 billion of them, and they're with you from the start of your life. Like all cells, neurons consist of several different parts, each serving its own function. It's a specialty and function. So let's look at this chart. You have a slide that has the structure of the neuron. Let's walk through its parts. So the outer surface of the neuron has a semi-permeable membrane. This membrane is essentially going to allow smaller molecules and molecules without an electrical charge to go through it, while stopping some of the larger or charged molecules. The nucleus of the neuron is located in the cell body. That's also called the soma. That cell body has branching extensions known as dendrites. Dendrites mean branches, like tree branches. So the dendrites serve as input sites where signals are received from other neurons. These signals are transmitted electrically across the cell body and down the major extension of the neuron called the axon, which ends at what we call terminal buttons. Those terminal buttons contain synaptic vesicles, or little fluid-filled sacs that have neurotransmitters. Those neurotransmitters are chemical compounds and chemicals that pass messages between neurons. Axons range in length from a fraction of an inch to several feet long. In some axons, glial cells form a fatty substance known as the myelin sheath, that's myelin, and it coats the axon, acting as an insulator. And where an axon is insulated, the speed of the action potential that neuron is able to send, it increases. The myelin sheath is not continuous, and there's little gaps that occur down the length of that axon in the sheath. These little gaps in the myelin sheath are known as nodes of Ranvier. And essentially that electrical impulse can skip from node to node, which is why the transmission can be faster on an insulated axon. A disorder of that myelin sheath that has to do with the hardening and breakdown of that myelin sheath around axons is known as multiple sclerosis. So let's review. Each neuron has a cell body, and from that cell body there are dendrites that connect with other neurons. They protrude their branches and they branch out to make other connections with other cells. From the cell body you'll have the axon, and the axon is a semi-permeable fluid-filled tube that transports essentially the electrical impulse down the length of the neuron. If that axon, that fluid-filled tube, if that axon is myelinated, then an electrical impulse can become quicker. It can go down the length of the axon quicker because it's going to essentially skip from node to node. At the end of the axon, you'll have multiple protrusions that connect with receiving neurons. That's the next neuron in the chain, in the communication chain. There's going to be terminal buttons, and in those terminal buttons, there's going to be things called synaptic vesicles. Those synaptic vesicles house neurotransmitters. And neurotransmitters are going to be the chemical messengers that are going to cross over to the next adjacent neuron and try to communicate about the action of the nervous system. Are we firing right now? Are we having electrical impulses in this area? Are we thinking about this thing, acting towards this thing? Is this happening now? Or is our body not firing an impulse? Are we resting? Are we disinhibiting the action potential? Are we exciting the system or inhibiting the system? Those chemical messengers are going to communicate that, the transfer of information from one neuron to the next. So now that we've talked about the basic structure of the neuron, let's talk about the role that these structures play in how neurons communicate with one another. We'll take a look at something called the action potential, the electrochemical signal that moves through the neuron and then jumps to the next neuron. Okay, as I mentioned, the neuron exists in a fluid-filled environment. There are fluids essentially inside and outside of the neuronal environment. Since it's semi-permeable, it means that really small ions and ions that don't really have an electrical charge, they can go in and out pretty freely. But the bigger ions, things like sodium and potassium and chlorine, and those ions carry an electrical charge. They have positive or negative charge. Those things, the semi-permeable membrane keeps in a certain status. And that status essentially is with the resting potential of the neuron being in a negative polarity. At rest, essentially, the ion gates and the special little doors, essentially, that allow these larger charged ions to go in and or out of the cell, those ion gates at rest are closed. And because those ion gates are closed, you'll essentially have a more negative charge on the inside of the axon than on the outside of the axon. Because there's differential charge by way of the molecules that are inside or outside of the cell at rest is said to be polarized. Polarized means essentially that it carries and creates a voltage. And at rest, the voltage is around negative 70 millivolts of a resting neuron. That polarization puts a cell in a state of readiness. It's negatively charged, kind of like the way that if you have a rubber band and you expand it back, it's ready. It's kind of ready to fling forward, right? So at rest, the neuron is ready and primed to do something. That state of readiness is what we call the resting potential. And in its ready state, those ions, those charged ions, they're going to line up on either side of the cell membrane. They're ready to rush across the membrane when the neuron becomes active and those ion channels open those gates. Those positive ions are ready to move into the areas where there's negative charge. So in a resting state, sodium with a positive charge is at a higher concentration outside of the cell. So it's going to tend to and want to move into the cell. Potassium, which also carries a positive charge, is more concentrated inside of the cell, and it wants to move out of the cell. So just like that rubber band stretched and ready to fire, ions of a positive charge or negative charge, they essentially, in their charged state, they line up on either side of the cell membrane, ready to equalize, ready to rush in or out in such a way as to go towards the direction of equalizing the charge. What this means essentially is that at rest, you're going to have some positively charged ions on the outside of the cell and a predominance of negatively charged ions on the inside of the cell. In the resting state, sodium, which carries a positive charge, is at a higher concentration outside of the cell. So it is going to want to and tend to want to move inside of the cell if those ion channels open up and allow it to do so. Potassium, which also carries a positive charge, is somewhat more concentrated inside of the cell. and its tendency will be to move out of the cell if those ion channels allow it to move out. And as I mentioned, the inside of the cell is slightly negatively charged compared to the outside due to the predominance of chlorine ions, which carry a negative charge. So in its resting state, the neuron has a negative millivoltage. It has a negative charge, around negative 70 millivolts. From this resting state, if a neuron receives a signal, then its state and its action are going to change. When a neuron receives signals at the dendrites due to neurotransmitters from an adjacent neuron binding to those receptors, the small ion channels, or those gates, they open on the receiving neuron's membrane. They allow positively charged sodium ions that want to go in because of charge and concentration differences to move inside of the axon, to move inside of the neuron. This influx of the positive sodium ions, it's going to internally change the inside of that cell to become more positively charged. If enough of those positive ions, by way of just the differential charge, if that charge reaches a certain level, which we call the threshold, the neuron essentially becomes active and its action potential or electrical chemical impulse is going to turn on. Going from the polarized state of a charge, polarized means differential charge, and at rest we know it's polarized, it has a negative charge. So when these ion channels open up, the cell becomes depolarized, less polarized. As more ion channels open and there's more depolarization, there is a massive influx of positively charged sodium ions and essentially a positive spike in the membrane potential, meaning its positive charge. This positive spike or depolarization constitutes the action potential if it reaches threshold. So something about the action potential, this neural impulse that you need to remember, is that it's like a light switch, and a traditional light switch. It's either on or off. It's not like a dimming light switch, which you can go from like light to medium to bright. It does not have a small, medium, or large action potential. If depolarization is enough to reach threshold, then the action potential will fire, all or none. This action potential is the electrical signal that typically moves from the cell body down the axon to the axon terminals. The electrical signal moves down the axon with the impulses jumping in sort of a leapfrog fashion between the nodes of Ranvier, if the axon is myelinated. At each point, some of the sodium ions that enter the cell diffuse to the next section of the axon, raising the charge past the threshold of excitation and triggering the next influx of sodium ions down the length of the axon. The action potential moves all the way down the axon in this way until reaching the end of the axon, its terminal buttons. And this action potential is an all or none phenomenon. This means, as I mentioned, that the incoming signal from another neuron is either sufficient or insufficient to reach that threshold of excitation or depolarization. There is no in between. There's no small, medium, and large. It's either on or off. Because it's all or none, the action potential is recreated or propagated at its full strength along every point of the axon. And you might think to yourself, wow, that's a lot of detail about one little cell. Well, this little cell is so foundational to how you behave and adapt to your environment. And the fact that it's so efficient and the fact that it's all or none means that you're able to be more efficient and more adaptive. And there is absolutely a restorative process after the action potential concludes. After an action potential has gone down the length of an axon, what's going to happen is those potassium ion channels, they're also positively charged. They're going to open, and those positively charged potassium ions are going to go out of the axon. Sodium had come in, and now potassium is going to go out. And when the potassium positively charged ions go out of the cell, it will return the axon to that resting potential of its negatively charged state. All right, so let's talk about what happens after neuron A has fired an action potential down the length of its neuron, down its axon, and the message has gotten to the end of neuron A, and it is trying to communicate with neuron B, which is the next one in the chain of communication. What happens at the end of the first neuron? When the action potential arrives at the end of the sending neuron, we'll call it neuron A, that action potential arrives at the terminal buttons. It has been, to this point, an electrical process. It's now going to become a chemical process because those synaptic vesicles that are holding the neurotransmitters, they're going to release those neurotransmitters, this is chemical now, these are chemicals, into the synapse or the synaptic cleft. The neurotransmitters are going to travel across the synapse and bind to receptor sites on the dendrites of the next neuron, the adjacent or next neuron. We'll call it neuron B. And that process, that action potential process, is going to repeat itself in the new neuron, assuming that that signal from neuron A is sufficiently strong to trigger an action potential, that there's enough depolarizing force. Once that signal from neuron A to neuron B is delivered, any extra chemicals, any extra neurotransmitters that are in the synapse, they're going to drift away. They're either going to be broken down into inactive fragments, or they're going to be reabsorbed into the sending cell in a process that's called reuptake. Reuptake essentially involves that neurotransmitter being pumped back into the neuron that had released it in order to clear out the synaptic gap. And clearing that synapse really has to do with providing a clear on and off state. But it also has to do with regulation of the production of those neurotransmitters. Clearing out the synapse essentially puts the neuron in sort of an off and on state. And from an efficiency standpoint, the reuptake process allows the sending neuron to know, oh, I've got enough neurotransmitters, I don't need to produce any more. So to review, at the end of neuron A that has sent an action potential down the length of its axon, upon receiving that message at the terminal buttons at the end of the sending neuron, that electrical impulse is going to be converted into a chemical signal. Those synaptic vesicles, those axon vesicles, they're going to release neurotransmitters into the synapse and the synaptic gap. And since neurotransmitters are fancy chemicals that have specific structures, think of them as like keys. You have keys to your house, you have keys to your parents' house, you have keys to your car, and all of those keys open different doors, right? Well, the same thing is true with neurotransmitters. They're like specific keys that can unlock specific doors. In this metaphor, the neurotransmitter is the key, and the receptor site on the receiving neuron is the lock. So when the receptor site on the neuron B, the neuron that's the next in the chain of communication, when that receptor site detects the presence of the correct key in its lock, it essentially has chemically received the message of what neuron A was trying to send. So let's talk a little bit more about neurotransmitters. So there are several different types of neurotransmitters released by different neurons. Much of what psychologists know about the functions of neurotransmitters comes from research on the effect of certain drugs, and especially the use of drugs for some psychological disorders. So neurotransmitters can be excitatory, inhibitory, or act in both ways. Most of your neurons essentially have one type of neurotransmitter. But since even one neuron is so highly interconnected with other neurons, that chemical message that's happening in the synaptic gap, it could have tons of different types of neurotransmitters in there. That's why the lock and key metaphor is a really apt metaphor for what's happening at the synapse. So let's quickly review some of these neurotransmitters, these chemical messengers of the nervous system. So neurotransmitters are specialized keys of the nervous system. They are specific chemical messengers that open up specific doors in the nervous system for different functions. They carry their information across one neuron to the next. And since each neuron often has one type of neurotransmitter that it releases, different neurons essentially are doing different things. Again, the lock and the key metaphor I think is very helpful. You can try to use your car key for your house, but it's not going to open your house door, right? So some of these neurotransmitters, they can try to bind with the receptor site on a certain neuron, but if the lock and key connection is not correct, the receiving neuron doesn't have to open its doors to act in that way. So when you think of neurotransmitters, you're going to think of them as being specialized chemical keys that unlock certain functions in certain neurons. They have certain actions. So let's talk about some of these specialized keys. Acetylcholine is a neurotransmitter that's involved in muscle action and memory. Its potential effect on behavior has to do with increased muscle action or increased arousal, and potentially increased or enhanced cognition. Black widow venom, therefore, increases acetylcholine levels. Botulism reduces the level of acetylcholine. And as you know from Botox, it essentially paralyzes the muscles, keeping them from moving. People that have Alzheimer's disease often have lower levels of acetylcholine, and this is implicated to some degree in cognitive skill, cognition, and memory. GABA is another neurotransmitter. It's implicated in brain function and sleep. A potential effect on its behavior might have to do with decreasing anxiety or decreasing tension. GABA is what your textbook talks about as a inhibitory regulatory brake pedal in the nervous system. It basically tells the brain, hey, let's slow down. Let's kind of ease off of firing so much. Let's regulate. Let's slow down. If GABA is the brake pedal, then glutamate is the gas pedal. It's an excitatory neurotransmitter. It's implicated in memory and learning and has potential effect on increasing learning and enhancing memory. It's the most prevalent neurotransmitter and often excites other neurons into firing. Next, we have norepinephrine, which is implicated in heart regulation, intestines, and how alert you are. Its potential effect on behavior has to do with increasing arousal or suppressing appetite. Part of the reason for this is that it's going to essentially decrease central nervous system firing, but increase peripheral nervous system function. So you'll often see that drugs can sometimes act on this system, like meth and cocaine. They act to increase norepinephrine levels. Norepinephrine also regulates sleep in conjunction with acetylcholine. So this is an important thing to remember, is that many of these keys sometimes work in tandem, either alongside or against one another. Another really important neurotransmitter is dopamine, and dopamine is implicated in mood, sleep, and learning, as well as voluntary movement. You'll see that the dopamine circuit and this discussion of multiple areas in your brain that are responsive to dopamine has to do with reward anticipation and pleasure sensation. Many stimulant drugs are going to activate those dopamine receptors, which is why those stimulants feel so good. Therefore, the potential effect of dopamine on behavior has to do with increased pleasure and suppressed appetite. Serotonin is another really important neurotransmitter implicated in the regulation of sleep, mood, and attention. Its potential effect on behavior has to do with essentially adjusting mood and regulating sleep. People with depression often have quite low serotonin levels. Therefore, many of the drugs that are given to people that suffer from depression will be selective serotonin reuptake inhibitors. That means that the serotonin stays in the synapse longer so it can act longer, so it can have more of an impact for a longer lasting time. Serotonin in the synapse means that it can fire the next neuron to tell the neuron, hey, we're okay, things are fine. Endorphins are involved in the pain and pleasure response. A potential effect on behavior has to do with decreasing anxiety or decreasing pain and tension. They are the natural opiates of the nervous system, and they decrease nervous system activity. And those neurotransmitters are often mimicked by opiates or opium or drugs that are of the morphine family. They mimic the effects of endorphins. And finally, we have oxytocin, which is a very unique and very special neurotransmitter because it is both a neurotransmitter that acts in the nervous system and a hormone that is used by the endocrine system in the bloodstream. It is both a hormone and a neurotransmitter. So it acts both upon glands in the endocrine system as a hormone and synaptic vesicles and synapses in the nervous system. And we also have some studies that show us that oxytocin is involved in the attachment process of trusting, belonging, and attaching to other people. All of these neurotransmitters are still being studied, still being understood, and there's more probably that we haven't even discovered or found yet. Now, when we talk about drugs and drug use, we know that drugs can interfere in some way with the action of neurotransmitters. Psychoactive drugs then can either act as agonists or antagonists for a given neurotransmitter system. Agonists are chemicals that mimic the neurotransmitter at the receptor site, like a key, a fake key that works like the same key. And an antagonist, on the other hand, blocks or impedes the normal activity of that neurotransmitter at the receptor. And that's like putting a piece of gum in the receptor site so that the key can't even get in there if it wanted to. Agonists and antagonists represent drugs that are prescribed to correct specific neurotransmitter imbalances in a person's underlying condition. So, for example, Parkinson's disease is a progressive nervous system disorder associated with low levels of dopamine. Therefore, a common treatment for Parkinson's disease involves using dopamine agonists, which mimic the effects of dopamine by binding to dopamine receptor sites, allowing those neurons to act. So what do we take away from all of that information? Well, we take away the fact that your nervous system is highly complex. It is tremendously interconnected with many pathways of neurons and nerve cells. It has to integrate afferent and efferent sensory and motor information. it grows, develops, and changes over the years. And one single neuron is not storing one memory or one thought or one habit. Those things are stored across an entire system of neurons and neural connections. So as we begin to learn more and more about how your nervous system acts and the action of neurotransmitters on the behavior of the human, we learn more and more about why people do what they do and how to help people do what they do more effectively and with more health. Okay, so how do we study the brain? Well, one of the ways that we can study the brain is by studying the brains of individuals that have had damage, pre-existing damage, traumatic damage to certain areas of the brain, and seeing how it's impacted their function. Another option is to actually lesion parts of the brain. This would be in animal models. They call it deep lesioning. And that's usually has to do with damaging certain neuron clusters or certain areas of the brain and then watching the animal to see how their behavior is impacted. We can also study the ongoing electrical impulses of the brain by EEG or even single axon recording of implanting an electrode and watching a specific cell, whether it fires or doesn't. Increasingly, however, we're more and more able to use information by using brain imaging techniques on individuals that haven't suffered any brain damage. So we can use X-ray, CT scan, a PET scan, an MRI, or an fMRI to assess different layers of analysis and layers of function in the human brain. Of interest to us, the most highly detailed function that we're able to see is with fMRI, or functional magnetic resonance imaging. fMRI operates essentially by showing live changes in brain activity over time by tracking blood and oxygen levels in the brain. It provides a more detailed image of the brain structure as well as better accuracy in real time. With that high level of detail, we can see which areas of the brain are active during different tasks and psychological processes. All right, so moving forward, I want to do a quick review of areas in the brain. This lecture would be 10 hours long if we went into each of them very, very deeply. I can definitely tell you that your textbook is a great reference here. I think it is at a good level of challenge, and I hope that it's going to inspire some inspiration and creativity and thought from you. So let's go through areas of the brain and get to know a little bit more about who you are on a brain level. Okay, so let's talk about the most internal, basic, and what Daniel Siegel would call downstairs part of your brain, the basic part of your brain. We're going to call that the hindbrain. It is in the most internal and deep area. It's located at the back of the head, and it looks like an extension of the spinal cord. It contains the medulla, pons, and cerebellum. So the medulla controls automatic processes, stuff within the realm of the autonomic nervous system, such as breathing, blood pressure, and heart rate. The pons, and that word means bridge, essentially serves to connect the hindbrain to the rest of the brain. It's implicated in sleep and arousal. The medulla and pons and some other structures nearby are known as the brainstem. And that aspect of the brainstem can span both the hindbrain, that's the lowest part, and the middle brain or midbrain. The other part of the hindbrain is the cerebellum. That's Latin for little brain, and it looks like a little brain. The cerebellum receives messages from your muscles, your tendons, your joints, and the structures even in your inner ear to control balance, coordination, movement, and essentially voluntary motor skills. The cerebellum is thought to be an important area for processing even some types of memories. In particular, procedural memory, or memory involved in learning and remembering how to perform certain tasks. So moving now into a more internal area of the brain, we have the midbrain. The midbrain is comprised of structures that are really deep within the brain, between the forebrain and the hindbrain. We have the reticular formation, which is centered in the midbrain, and it actually extends up into the forebrain and down into the hindbrain. That reticular formation is super important in regulating sleep and wakefulness, general attention and arousal, alertness, and even motor activity. You'll also have the SN and the VTA that are located in the midbrain. Both of those regions contain cell bodies that produce the neurotransmitter dopamine. And both of those areas are critical for movement. So degeneration of the SN and the VTA are involved in Parkinson's disease. And those areas, since they're involved in the dopamine circuitry, they're involved in mood, reward, and even addictive action. Now moving towards the outermost level of the brain, we have the forebrain. And the forebrain is going to have many different areas. Let's start with the limbic system. So the limbic system is involved in processing emotion and memory. It's kind of cool. That sense of smell and your sensation of smell, it actually projects directly onto the limbic system, which is why smell can evoke such a strong emotional response in you. The limbic system is made up of a number of different structures, but we have some structures that are particularly important to note. We have the hippocampus. So the hippocampus is an essential structure for learning and memory. We also have the amygdala, and that means almond because that's what it looks like in both hemispheres of your brain. And the amygdala is involved in our experience of emotion and essentially tying emotion to our own memories and connections. It has a lot to do with negative emotion like fear responses and is implicated in emotional awareness and emotional expression. You'll have the thalamus as well that's going to serve as a relay station for a lot of our sensory information. The thalamus is at the top of the brainstem and it's sorting out info and trying to send it to appropriate areas in the brain so that those areas can interpret the sensory information and respond accordingly. In the forebrain, you'll also have the basal ganglia, which has to do with working with the cerebellum to deal with control and coordinating motor movements, especially voluntary movements like walking and dancing and moving. And you'll also have the hypothalamus, or the body's internal thermostat, that has to do with regulating a number of homeostatic processes, including body temperature, appetite, blood pressure, but also serving as an interface between the nervous system and the endocrine system in the regulation of sexual motivation and sexual behavior. Since it's implicated also in just the overall regulation of your body, it's going to have some say over emotion, stress, and rewarding situations. There are 4 lobes of the brain, and those 4 lobes are frontal, parietal, temporal, and occipital. The frontal lobe is located in the forward part of the brain, and it extends back to a fissure known as the central sulcus. The frontal lobe is involved in reasoning, motor control, emotion, and language. At the very back of the frontal lobe, you have the motor cortex. And that cortex, or part of your brain, is involved in planning and coordinating movement. At the very front of your frontal lobe, you have the prefrontal cortex. And that is a super important area that's responsible for what we call executive function. That's higher level cognitive functioning. The frontal lobe also includes Broca's area, which is essential for language production. Moving back from this area, we have the parietal lobe. So the brain's parietal lobe is located behind the frontal lobe. It's involved in processing information from the body's senses. Therefore, at the very front of the parietal lobe, you'll have something called somatosensory cortex, which is an essential brain region for processing sensory information across the body, including stuff like touch, temperature, and pain. Your book shows that the somatosensory cortex is organized kind of like a map, which means that there are spatial relationships that exist in the body that are generally maintained on the surface of the somatosensory cortex. So on that somatosensory cortex, you could have, this is where the ankle is, this is where the fingertip is. If I stimulate this area, it's the elbow. We call that point-to-point mapping. And that exists both in the motor cortex of the frontal lobe as well as the somatosensory cortex of the parietal lobe. Underneath the parietal lobe, we have the temporal lobe. And the temporal lobe is located essentially on the side of the head, near the temples. That's why it's temporal. We can kind of think near the temples. It's associated with hearing, memory, emotion, and some aspects of language. The auditory cortex, which is the main area responsible for processing auditory information, is located within this temporal lobe. Wernicke's area is an area important for speech comprehension, is located here within the temporal lobe. And at the very back of the brain, you have the occipital lobe. The occipital lobe has the primary visual cortex, and that's the area of your brain that's responsible for interpreting and integrating incoming visual information. That occipital cortex, the part of your occipital lobe, it's actually organized by the retina, which means that there's a point-to-point representation and a close relationship between the position of an object on your visual field and the position of that object on the visual cortex. Now, much of what we know about lateralization of the brain, lateralization means that right and left hemispheres are specialized for function. We know from split brain research. Now, your brain and those four lobes are lateralized or specialized within two different hemispheres, the left hemisphere and the right hemisphere. In general, left and right hemispheres work together, not by themselves, but together to process the world and respond to the world. That being said, parts of your brain in left and right might be quicker to do certain tasks than the other side might. Why would you want a brain that was specialized or lateralized in its function? Well, think about this. Let's say that you are a boss of 100 people. You're in charge of like Habitat for Humanity. You're going to build homes. A good boss is going to have these 100 people work together on different tasks to build this house. Some people are going to get supplies. Some people are going to do the foundation and the cement. Some people are going to build and saw 2x4s and hammer nails. Other people are going to paint and get windows, right? So if you have 100 people and they're all doing different tasks that are towards the main goal of building the house, that house is going to be built much quicker than if all 100 people do the same task at the same time. Imagine how inefficient that would be if all 100 of your people were doing all of the tasks. So you were all hammering one nail at the same time. It doesn't even make any sense. Therefore, your brain is specialized. It's efficient. It's able to deal with a multitude of inputs and get multiple things done well at the same time. Broadly speaking, the left hemisphere has an aptitude for language and speech, both written and spoken. You'll find both Broca's area and Wernicke's area in the left hemisphere. In the right hemisphere, you'll see more nonverbal information, spatial perception, visual recognition, emotion, and faces. So they're specialized. This means that they do those tasks a little bit better and a little bit quicker than the other side does. But if the brain is damaged or there's some problems with certain areas, the brain also has the ability to communicate and connect and make up for some losses or disfluencies. One of the ways that it makes up for stuff is through the corpus callosum, which is the band of neurons and fibers that connect the two hemispheres of the brain. And all in all, you guys, I want you to walk away from this chapter not being bogged down by a lot of individual brain parts that do certain things, but to understand the gravity and the elegance and the complexity of your brain. It's amazing. It has the ability to grow and connect and heal. Even with damage, it has the ability to reorganize and reconnect. And even areas that are not specialized for a function can take on some function. It shows neuroplasticity, which is even the growth of neurons. It can show substitution of function or even growth of axons. Your brain's pretty awesome. And since you're listening to this right now, I think you're pretty awesome. Okay, so the end of this chapter is going to talk about the endocrine system and some genetic considerations. I'm not going to belabor the point because I think that the textbook does also a very good job of walking us through this, but let's walk through a little bit of the endocrine system and why we care about it in light of biological psychology. Okay, so the nervous system is a quickly acting system that's based on an electrical chemical impulse. We know this is the neuron, the action potential, and the synaptic transmission. The endocrine system also communicates in the body, but it communicates by way of the bloodstream. Slower process by way of bloodstream transmission. So in the nervous system, you have neurotransmitters, and in the endocrine system, you have hormones. So essentially, we're going to have a set of glands that are going to release certain hormones. And those hormones are going to be chemical messengers that are in the bloodstream and interconnect with the nervous system. So these two systems, endocrine and nervous system, are going to be interconnected and communicating with one another. And you can think about it this way. Well, there's lots of things in your brain, neurotransmitters, brain structures, nervous system function, as well as your endocrine system, your hormones, and the things that are going on in your body that can lead you to behave, feel, or think in certain ways. Now, this isn't meant to overwhelm you. This is meant to make you be in awe of how complex you're organized. Also, we see in this chapter a discussion of genetic inheritance. I think there's a really cool place in the text about twin studies, identical twin and fraternal twin studies. Make sure you take a look at that and read that paragraph. I think you'll enjoy it quite a bit. We're learning more and more about why people do what they do through studying our genetics or our genes. So our genotype has to do with our genetic inheritance. But it's not static. It interacts with our actual environment, nurture, and experience over time to alter its expression or phenotype, how we actually display our behavior and display our characteristics. So who you are is not just a genetic or biological nature. It is also the environment in which you're placed. So what's a good takeaway from this? Don't ever walk out of this chapter thinking that, well, my brain is that way and it's always going to be that way and that's that. Or my genetics from my family, they're just like this and it's just going to stay this way. You're not set in stone like that. The environment and nurture have a lot of influence as well. You are who you are with nature, genetics, and nurture, experience. genes and environment work together to determine outcome. Sometimes you can't change the biology of what's going on for you or what is strong or weak for you, but you might be able to change the environment under which you're experiencing those things. So as you prepare and study and you've got tests coming up and as you have new pressures and new concerns, please, please, please choose environments that are going to help you succeed. Calm, safe, healthy environments. Okay, you guys, I think I've adequately parented you a little bit here. You did so good in this lecture. I'm so, so proud of you. I know all of the things on your to-do list sometimes might seem a little bit difficult to handle, but I just want you to know I'm proud of you and I'm so glad to be your professor. So where are we going from here? Well, in the next chapter, we're going to be talking about sensation and perception. We're going to expand this biological approach into the realms of sensory and perceptual experiences. If you have any questions about due dates or homework or discussion posts or things that are going on, you're going to reach out to me. Otherwise, I'm proud of you. You did a great job. Congrats, everybody. I'll catch you next time.